Hello everyone, welcome to Life of Fusion, where knowledge meets excellence. Myself, your educator, Dr. Shashwat Kumar Mohanty, and in this video, I am going to talk with you about how to solve problems on linkage mapping. The linkage mapping is one of the very important part of Unit 8 of CSIR-NET syllabus. And every year, you must get around six questions in part C of unit 8, and this part is one of the most important part because at least one question must you'll get from linkage mapping. So that's why this part is very important for those who are preparing for CSR net life sciences. So the linkage mapping or the mapping of gene means here you are mapping between genes. So if I say there is two gene or three gene, for example, gene A, gene B, and gene C, we are searching, we are finding the distance between these three genes. So these genes, the distance between these genes are mostly calculated from the recombination frequency. Okay, so if you don't know what is recombination frequency and how to calculate recombination frequency, you can have a look on our previous video where I also uploaded a video on how to calculate recombination frequency. As I told you in my last video that the genetic mapping or recombination frequency, okay, this mapping, it's not exactly a physical mapping. In a physical mapping, you used to find the distance between gene in the means of base pair. Okay, but here we'll find mostly you'll find the order of the gene, and the genes are close or far apart. Okay, so based on we here in this mapping method, we this method just give you a approximate mapping. Okay, means as you know about linkage and crossing over, if two genes are close together, then the chance of crossing over will be less. If the two genes are far apart, the chance of crossing over will be more. So if there is a crossing over, means the chance of production of recombinants offspring will be more. Recombinants. So here basically we are finding the percentage of recombinant offspring produced between two genes. If two genes they produce more number of recombinant frequency, means the two genes are far apart. If the two genes will produce less number of recombinant offspring, then these two genes are close together. So that's why this is the percentage of recombinant offspring produced between the two genes. Okay, so that's why the genetic mapping or linkage mapping is directly proportional to the recombination frequency. Okay, so linkage mapping is directly proportional to recombination frequency, means the number of the what you'll get in recombination frequency, that is directly equal to the linkage mapping. Okay, so that's why 1 unit of map distance is equivalent to 1% of recombination frequency. So if you get a recombination frequency of 10%, that equal to the gene distance between the genes are 10. So to give this distance a particular unit, it was used by the name of Morgan as centimorgan. Means if two genes produce 10% of recombinant offspring, means the two genes are 10 centimorgan apart. If two genes will produce 20% of recombinant offspring or 20% of recombination frequency, it means the two genes are 20 centimorgan apart. Simple. So here basically you are finding the distance between the gene in the means of recombination frequency. So what will be the formula for the distance between the genes? So the distance between these genes formula will be directly as similar as the formula of recombination frequency. So the map distance will be number of recombinant offspring produced. How many number of recombinant offspring produced divided by total number of offspring into 100. As I mentioned in previous video, when you are going to find the recombination frequency in the F2 generation, you need to get around more than 1000 number of offspring. If and from that 1000 number of offspring, you have to calculate how many offspring are having recombination. Means there will be a crossing over. So that's why simple we are calculating the percentage. So x can be a number of recombinant offspring, and maybe if 1000 is your total number of offspring into 100. Okay, into 100 means just we are calculating the percentage. So this formula is directly proportional to the formula of recombination frequency. For a part B question, you can keep in your mind that the genetic mapping was constructed in 1911 by Alfred. Okay, so there are two types of genetic mapping. One is mapping between 2-point cross and mapping between 3-point cross. So mapping between 2-point cross means mapping between two genes. For example, gene A and gene B. So here you are calculating that what is the recombination frequency between these two genes. Okay, directly we get the recombination frequency. But in three genes, okay, gene A, gene B and gene C, we need to find the recombination frequency between all the A to B, B to C and A to C. Okay, so before that, in 3-point cross, we have to find the gene order. Okay, and in CSIR exam, the mostly important questions come from the 3-point cross. So that's why in this video I focused on the questions how to solve problem from the 3-point cross genetic mapping. Okay, so, as I told you, this mapping is calculated between the distance between linked gene. Okay, linked gene means in the linked gene, the chance of recombination frequency will be less. Okay, it means they are in a particular chromosome, not in different chromosome. Are you getting it? They will be in a particular chromosome and they should be linked. Okay, and that should not be too much far away. Okay, and I told you that the previous video, the recombination frequency should not exceed 50%. Okay, if the exceeding 50% means these genes are very far apart, means they are not linked. When they are not linked, we are not going to calculate the genetic mapping. Okay, so if the two genes are far apart, means what? The recombinance offspring produced or the crossing over and the recombination will be very high. Means the genetic mapping will be more when these two genes are far, the distance will be more. If the two genes are close together, means the chance of recombination will be less. Means they are, you can say they are linked gene. Okay. So, based on this, first come to how to solve problems on 3-point cross of genetic mapping problem. Okay, 3-point cross means they based on the three loci. Okay, three loci, they are segregated by two alleles. You know, there are two alleles for each loci. Okay, so as you know, a double crossover product separate the gene in the middle. So here, why this double crossover product? In the 3-point cross, we have to cross, you have to find the distance and order between the three gene. When you find the order between the three gene, we have to find the middle gene. That is very essential for these types of problem. Okay, so, if you want to find a distance between or order of this gene, you have to take, as it mentioned in previous video, one parent should be homozygous dominant, one parent should be homozygous recessive. Like I can say, one parent will be kept. Maybe if genes are A, B, C genes, okay, then the parent one will be capital A, capital A, capital B, capital B, capital C, capital C, and parent two will be small a, small a, small b, small b, small c, small c. Here in this case, as both are dominant and both are recessive, I can take as parent 1 as capital A, capital B and capital C, and parent 2 as small a, small b, small c. Okay. What is parent 2? Parent 2 will be a tester as I mentioned in your recombination frequency video. So, this is parent 1, this is parent 2. So, to find the distance between these genes, you have to cross between the parent 1, which is your F1 hybrid, okay, and parent 2, which is your homozygous recessive. When you cross between these genes, okay, then we will get some of the offspring which don't show any recombination. Okay, When the offspring don't show no recombination, no recombination. There is no recombination means those offspring you can say they are parental type. Okay, parental type. No recombination means they are exactly same as the parent. If they have recombination means they will be your recombinant offspring. They are your recombinants. So if there is three gene A, B, and C, there is chance that there could be a single crossover between any of these gene. Okay, and there could be a double crossover as you can see in this in this figure. Okay, so here as I mentioned that the gene order is A, B, C. This is for an example I am giving here. So this is the gene order A, B, and C. Okay, so B is the middle gene. So if you cross between A, B, C, capital A, capital B, capital C, and small a, small b, small c, so what you will get? If there will be a crossover between B and C gene, okay, so the product will be capital A, capital B, and small c, then small a, small b, and capital C. Similarly, if there will be a single crossover between gene A and B, then what will be the product? Capital A, small b, small c, then small a, capital B, and capital C, small a, capital B, capital C. Are you getting it? So this you have to find after you get the gene order. So how will get gene order? You will get gene order from the double crossover product. Okay, this is your double crossover product. So where as I mentioned that the B gene is the middle gene. So as you see here from the double crossover product, the products are capital A, small b, and capital C. If you cross between capital A, capital B, capital C, small a, small b, small c, if there will be a double crossover product means crossover between A gene and B gene. Also there will be a crossover between B gene and C gene. So what do you get here? I'll write this one. This is very important. Capital A, small b, and capital C. Then small a, capital B, and small c. Small a, capital B, and small c. As you can see from the double crossover product, you can easily find out the middle gene. Okay, see here in the first product and this is the second product. In the first product, capital A and capital C are from parent 1, whereas small b is from parent 2. Similarly, in second types of product, the small a and small c are from parent 2, whereas capital B is from parent 1. What you saw here, in double crossover product, the two genes will be from one parent, whereas the other gene will be from other parent. Are you getting it? In double crossover product, two genes from one parent, and the third gene, other gene, from other parents means what? The gene which is from came from other parent, that is the middle gene. As you can see here in this product 1, small b gene, it came from small b from parent 2, whereas in product 2, capital B gene, if it comes from parent 1, so B gene is the middle gene. Okay, so we'll solve this through a problem. So here you get a calculation problem. The problem is for a gene A, B, and C, the first crossover between the homozygous dominant and homozygous recessive, okay, they got the F1 hybrid. This is the example of recombination frequency. You will get directly questions like this. Okay, so after the F1 hybrid, we test cross, the tester will be homozygous recessive. These are the F2 offspring. We will get the F2 offspring. So from the F2 offspring are the most important type. So in the F2 offspring, then the question is that in the F2 offspring, they mention the genotype. You can see A, B, C, small a, small b, small c, small a, capital B, small c, and we are the number of observed offspring. How many means what? How many offspring showed this genotype? How many? Okay, so 390 showed A, B, capital A, capital B, capital C. 374 showed small a, small b, small c. Okay, so here the first step is we have to find which of the following genotypes are parental type. Mean exactly same as parent. Mean there will no recombination happen in between the genes. Means they are parental type. And what are the double crossover product? Double crossover product means from the double crossover product you can easily find the what? Order of a gene. Order of these three genes. Gene A, gene B, gene C. Okay. So how to find for a for parental type the highest number of offspring, okay, maximum, because these are linked genes, it means the chance of crossing over will be less. So the parental type will be number of offspring. They will show the parental type will be very high. So the highest number of, when you get the question, the highest number of offspring which will show the particular genotype, that genotype is directly your parental type. There, as you can see, capital A, capital B, capital C, small a, small b, small c. These are having 390 and 374 number of offspring. Means these two genotypes are parental type. Now we got parents. Okay, initially we didn't get parents. Now we get parents. Parent 1 is capital A, capital B, capital C. Parent 2 is small a, small b, small c. Then double crossover product. As you know, okay, if there will be a crossover between one genes, if I write small capital A small a, capital B small b, capital C small c. If there will be a crossover between these two genes, this crossover is to inhibit the crossover between the nearby genes. Okay, remember this one. Okay, if there will be a crossover between one gene, that crossover will try to what? Limit or inhibit the crossover between the nearby genes. That's why the chance of double crossover product will be very less. Okay, this inhibition is called as interference. I'll upload a video on this interference, coincidence and interference. After interference, how many percentage will show the coincidence? Means they will show the double crossover of product. Means the chances of coincidence or the chances of production of double crossover product will be very less. Means what? The number of offspring they will show the double crossover product will be very less. In the lowest number of offspring which you will find, that is the double crossover product. So as you can see here, okay, capital A, capital B and small c, small a, small b and capital C, these are the double crossover product. As the number of offspring they showed this type of genotype is 5 and 8. Okay, then what is the next step? We have to find what? The gene order. What is our double crossover product? Capital A, capital B, small c. Then small a, small b and capital C. What are the parents? Parents are capital A, capital B, capital C, small a, small b and small c. As you see here, capital A, capital B from one parent, this is parent 1, this is parent 2 and small c from other parent. In this case, small a, small b from one parent and capital C is from other parent. So what is the middle gene now? The middle gene is C. C is the middle gene. Means what is the gene order? Gene order. So gene order is A, C, B. Okay, A, C, B is the gene order. Okay, now you have to find the single crossover product. Now you know ACB is the gene order. So if ACB is the gene order, if there will be a single crossover between C and B, so what will be the product? Capital A, capital C, small b. Okay, capital A, capital C, small b means I mentioned here single crossover product between C and B. Then small a, small c, capital B, small a, small c, capital B. That is your single crossover between C and B. Similarly, if you have single crossover between A and C, what will be the product? Capital A, small c, small b. capital A, small c, small b. Then small a, capital C, capital B. Small a, capital C, capital B. That is your single crossover product between A and C. Okay, are you getting it? So now here in this case, the most important thing is to find the parental type, then to find the double crossover product. From the double crossover product, you can easily find the what order of a gene. Then you can easily find the single crossover product and double crossover product. So now, if you are going to find, we have to find the first distance between A to C, then distance between B to C. Okay, or you can say C to B, whatever. So now, EC, we have to find the crossover product between A to C. Here you are going to find the distance between A to C means what? The crossover product means the recombinant offspring produced between A to C. So, how many number of recombinant offspring will produce from A to C means what? The single crossover product between A and C, and also the double crossover product. When in double crossover also we have a what recombination between A to C. So what will be the formula here? The map distance is, so number of single crossover between these genes plus total number of double crossover product divided by total number of progeny into 100. Means what? This completely indicates as number of recombinants between these two genes. Means what? In a single crossover product, also we have the recombinants between the two genes, and also in the double crossover product, we have the recombinants between the two genes. That's why number of single crossover product between these genes plus total number of double crossover product divided by total number of progeny into 100. So first find the distance between A to C. So single crossover between A to C is 80. Number of offsprings are 81 plus 85. So 81 plus 85 plus 8 plus 5 13. That is your double crossover product divided by total number of progeny is your 1000 number of progeny into 1000, sorry, divided by 1000 into 100. So we'll get 179 divided by 1000 into 100, that equal to 17.9 centimorgan. Similarly, when I'm going to find the distance between B to C, means number of offspring, so single crossover between B to C, 27 plus 30 plus total number of double crossover product 8 plus 5 13. We will get 70 by 1000 into 100, that is equal to 7 centimorgan. So distance between A to C is 17.9 centimorgan, whereas distance between B to C is 7 centimorgan. So gene order is ABC. A to C is 17.9 and B to C is 7. So distance between A to B will be common. 17.9 plus 7 will be 24.9 centimorgan. Okay. So here I have gave you questions. One question. So give your answer option A, B, and C. This down are your answer. Give your answer in the comment box. Okay. So also I'd like to announce that we are going to start a free webinar series. Free webinar series for CSIR-NET Life Sciences. Okay. So those who are interested, you can find the link in the description. Okay, find the link in the description. In the description, I have provided a Google form link. Fill the Google form link if you are interested to join the free webinar class for CSIR-NET Life Sciences. Thank you, everyone. Subscribe to my channel and try to share with your friends. Thank you, guys.